Where does life begin? For generations, we have been taught a single intuitive story. We picture a warm, shallow pond bathed in gentle sunlight. A primordial soup, as Darwin mused, energized by the sun and lightning. It is a story of creation that fundamentally depends on light. This assumption has guided our entire search for life, both on Earth and beyond. We search for the Goldilocks zone around distant stars. Planets where liquid water could exist on the surface, touched by solar energy. But what if this assumption is wrong? What if our entire search has been looking in the wrong place? What if the cradle of life wasn't a warm, shallow pond, but rather a place of unimaginable pressure, total crushing darkness, and extreme toxic heat? This is the story of an idea that could fundamentally rewrite our own origin. It suggests our deepest ancestors were not born of the sun, but forged in the violent chemical fires of the deep earth. Now, let's dive into the data and uncover the reality hidden in the deep. For most of scientific history, this solar-centric view of life wasn't a hypothesis. It was a biological law. The equation was simple: no sunlight, no photosynthesis. No photosynthesis, no primary producers to form the base of a food web. Therefore, no sunlight meant no complex, abundant life. The deep ocean, far below the sun's reach, was logically classified as a biological desert. We expected to find only a sparse rain of dead material, marine snow, supporting only the most desperate and specialized scavengers. This was the established, undisputed, normal world of biology. And then, the year 1977. The Deep Submergence Vehicle Alvin, operated by the Woods Hole Oceanographic Institution, was not on a biological mission. Geologists were using it to explore the Galapagos Rift, nearly 2.5 kilometers down. They were hunting for geological heat, not for life. The mission plan was to find predicted hydrothermal vents, cracks in the seafloor, where cold seawater would seep into the Earth's crust, get superheated by magma, and jet back out. The Alvin descended into the icy, crushing blackness of the abyss. The temperature outside the submersible hovered just above freezing. The pilots navigated a barren, volcanic terrain. And then, a signal. The external temperature probe suddenly jumped. The water outside was no longer near freezing. It was warm. Through the viewports, the team saw something that simply could not exist. First, shimmering, milky blue water, hazy with chemicals. And then, emerging from the darkness, the source. It was not a barren desert, but a dense, thriving oasis, clustered around fissures of billowing, mineral-rich water. There were fields of giant clams over a foot long. Dense colonies of ghostly white crabs and mussels. And most bizarre of all, forests of giant tube worms, 6 feet tall, with blood-red plumes. This was not a sparse, starved ecosystem. This was a vibrant, bustling city. The scientists on board were stunned. They had discovered a complex ecosystem thriving in total darkness, isolated from the sun. The discovery shattered the foundations of biology, demanding an answer to a single profound question. If not sunlight, what was powering this? The answer was not found in the visible creatures, not in the giant clams or the blind crabs. It was discovered in the fluid itself. When we finally sampled this black smoke, we found it was a toxic, acidic brew. When we finally sampled this black smoke, we found it was a toxic, acidic brew. It was scorching hot, stripped of oxygen, and saturated with metals. And critically, it was loaded with a chemical that is profoundly toxic to most complex life on Earth: hydrogen sulfide. The unmistakable smell of rotten eggs. To us, it is a lethal poison. But to the life thriving at these vents, we discovered it is the very fuel of creation. The true primary producers of this dark world were invisible to the naked eye. They were not plants, but vast, thick mats of extremophilic bacteria and archaea. These ancient microbes had evolved to do something truly impossible. They had tapped into a completely different power source. They were harnessing the raw geothermal energy of the Earth itself, captured in the chemical bonds of the sulfide. We gave this revolutionary process a name: chemosynthesis. It is the recipe for life without light. Let's deconstruct how this chemical engine actually works. In the sunlit world, photosynthesis uses the energy from light to power a reaction that stitches carbon dioxide and water into sugars, the basic fuel for life. Chemosynthesis accomplishes the exact same goal. It builds sugars from carbon dioxide. But instead of using light energy to drive the reaction, it seizes the chemical energy locked inside that toxic hydrogen sulfide. The microbes effectively burn the sulfide with oxygen from the surrounding seawater. They capture the energy released from this reaction and use it to run their own internal factory, fixing inorganic carbon from the water into the organic matter that forms their bodies. They are creating a mountain of biomass, not from the sun, but from geology and chemistry. This is the base of the entire food web. These bacterial mats become the grasslands of the abyss. The ghost white crabs, the shrimp, and the mussels all graze directly upon them. They are the primary consumers of a food chain completely divorced from the surface. But this discovery revealed an even more elegant and bizarre solution. The most iconic vent animal, the giant tube worm Riftia pachyptila, takes this one step further. When we first studied these 6-foot-tall creatures, we were profoundly baffled. They had no mouth, no stomach, no digestive tract at all. How could they possibly eat? The truth, we learned, is that they don't. They farm. The vast majority of the tube worm's internal body cavity is a specialized organ, the trophosome, which is packed densely with symbiotic chemosynthetic bacteria. This is not just an animal, it is a cooperative superorganism. Its bright red plume, fluttering in the vent current, is not for feeding. It's a complex, hyper-efficient gill, absorbing 3 ingredients simultaneously. From the ambient deep seawater, it pulls in oxygen. From the toxic vent fluid, it pulls in hydrogen sulfide and carbon dioxide. A unique, specialized hemoglobin in its blood binds all 3 and transports them safely to the bacteria waiting inside. The bacteria are given a safe home and a perfect, steady delivery of all the ingredients they need. In return, they perform chemosynthesis inside the worm's body and leak the sugars and nutrients they create, feeding their host directly from the inside out. It is a perfectly efficient, self-contained biological factory, running on toxic gas miles below the sun. The discovery of chemosynthesis was more than just a new line in a biology textbook. It was a fundamental shift in our understanding of life itself. It demonstrated unequivocally that life was not a slave to the sun. It proved that life is a profound chemical inevitability, capable of arising and thriving anywhere a source of energy and a few key ingredients are present. This realization immediately forced a far more profound question. If chemosynthesis can sustain such a rich ecosystem down here, could it also be the place where life began? Suddenly, Darwin's warm little pond had a serious, violent competitor. These hydrothermal vents, we realized, are not just oases. They are ideal chemical reactors. They possess all the necessary components for abiogenesis, the origin of life from non-life. First, they have a powerful energy source. Not lightning, but the steep chemical and thermal gradients. The sharp energetic difference between the hot, reduced vent fluid and the cold, oxidized seawater. This gradient is a natural battery, providing the spark. Second, they have a concentration of the raw materials. Methane, ammonia, hydrogen, carbon dioxide, and phosphate, all the building blocks of RNA, DNA, and amino acids concentrated in one place. And third, they possess the perfect infrastructure. The porous, mineral-rich structure of the vent chimneys are filled with countless tiny iron and nickel-rich pores. These microscopic pockets act as natural test tubes, or catalysts. They trap and concentrate these simple molecules, forcing them to interact, driving the reactions that could, over time, build the first simple metabolic pathways, and eventually, the first protocells. This wasn't just a habitat, it was a plausible engine for creating life from non-life, a blueprint for genesis powered by the Earth itself. This new, radical hypothesis that life began in the deep, volcanic dark was transformative. But the true power of this discovery, its elixir, was not just what it told us about our past. It was what it told us about where to look for life, right now, beyond Earth. For decades, the search for extraterrestrial life, astrobiology, was crippled by the Goldilocks assumption. We needed to find a planet just like ours, at the perfect distance from its star. One that was not too hot and not too cold, so that surface liquid water could exist. By this metric, our own solar system looked barren, with Earth as its sole miracle. But the vent hypothesis changed the equation. It proved that life doesn't need a star. It just needs liquid water, a few simple chemicals, and a source of energy. And that energy source, we now know, can come from a planet's own internal heat. Suddenly, the potential for life in our solar system exploded, not on the sun-baked surfaces, but in the hidden liquid oceans of the outer moons. Consider Jupiter's moon, Europa. From the outside, it is a dead, frozen wasteland, encased in a shell of ice miles thick. But we know from magnetic field data and orbital mechanics what lies beneath that ice. A vast global ocean of salty liquid water containing more water than all the oceans of Earth combined. We also know that Europa is constantly being kneaded by the colossal gravity of Jupiter. This tidal friction generates enormous heat in its rocky core. What happens when a hot, rocky core is in direct contact with a global liquid water ocean? The conclusion is inevitable. Europa's seafloor must be covered in hydrothermal vents. It possesses all the same ingredients we found at the Galapagos Rift. It is a dark, chemical reactor, completely independent of the sun. It's a perfect environment for chemosynthesis to arise and sustain a biosphere. The implication is staggering. The most promising place to find active living aliens in our solar system is in an environment that is a perfect mirror of our own deep ocean. And Europa is not alone. Look to Saturn's tiny moon, Enceladus. The Cassini spacecraft, in one of the great discoveries of our time, flew directly through the plumes of water, jetting into space from tiger-striped cracks in its icy shell. It sampled that water. What it found was a profound confirmation of the vent hypothesis. The plume contained salt, confirming a liquid ocean. It contained silica nanoparticles, which only form in hot water above 90 degrees Celsius. Conclusive proof of active, ongoing hydrothermal vents. And most critically, it found molecular hydrogen and methane. This hydrogen is the smoking gun for chemosynthesis. It's a powerful chemical fuel, the equivalent of a free lunch for any microbe that can metabolize it, just as our vent microbes metabolize sulfide. The discovery of chemosynthesis, born from a geological mission in 1977, has become the single most important astrobiological blueprint we have. It gave us a new way to see, reframing frozen, distant moons as the most compelling targets in our search for a second genesis of life. It taught us that habitable doesn't mean like Earth's surface. It means like Earth's deep sea. This journey to the deep, dark oases of Earth's sea floor and to the hidden, speculative oceans of icy moons. It fundamentally redefines our own place in the cosmos. The hydrothermal vent hypothesis is a powerful counter-narrative to our sun-drenched origin story. It suggests that our oldest, most fundamental story, our creation myth, born of light and air, may only be a recent chapter, and that the prologue was written in total darkness and crushing pressure. We are still deciphering this ancient text. The scientific debate between the warm little pond and the deep, dark vent continues, because this is a question that cuts to the very core of our own identity. Who are we? Are we the improbable, fragile miracle of a sun-baked surface? Or are we the inevitable, resilient, and resourceful children of a planet's own inner fire? The discovery of chemosynthesis, the elixir from this journey, suggests the latter. It paints a picture of life not as a lucky, delicate accident, but as a fundamental driving impulse of a geologically active world. Music. It tells us that when a planet is truly alive, with a beating, molten heart and a circulating liquid ocean, life itself may be an unavoidable consequence. From the first simple protocell clinging to a mineral pore in a chimney wall, to the complex ecosystems of riftia worms, farming bacteria in the dark, and billions of years later, to the consciousness that builds a submersible to return to that very spot. The story of life on Earth may not have begun with a glance up toward the sun. It may have begun with an awareness of the deep chemical energy percolating up from the Earth itself. Our deepest, most ancient ancestry, it seems, is written not in sunlight, but in stone, in pressure, and in the profound generative darkness of the deep.